I would like to introduce her very short. Aviv Regev is the head of genetic research and early development. Prior to Genentech, Regev served as chair of the faculty, core member, and founding director of the Klarman Cell Observatory at the Broad Institute of MIT and Harvard, and as professor of biology at MIT and investigator at the Howard Hughes Medical Institute. And most important, she is a founding co-chair of the Human Cell Atlas. Among many honors, she is an elected member of the US National Academy of Science and National Academy of Medicine. And now, for her outstanding research, Professor Aviv Regev is also awarded with the Ernst Schering Prize 2021. We are honored to have Aviv Regev via stream here from California. A warm welcome again from you here in the Leibniz Saal. And why single-cell genomics are like a fruit salad and not like a smoothie, you will discover when you listen to Aviv Regev's lecture. She will explain us why the Cell Atlas is a roadmap to in health and disease. Aviv Regev, the screen and the Saal is yours.
Much. I hope everyone can hear me. It's a great honor. I wish I could be there in person, but if there's something we learned in the last year and a half is that our plans can be thwarted and we can still make it happen in some way. So I'm extremely grateful for the foundation for the award, but I'm also very grateful to Maren and Katya and others who have made this work in unprecedented time. So I'll start with my gratitude for the prize in honor of Ernst Friedrich Christian Schering, as well as to the Schering Foundation. And I know that the foundation is committed to the interface between science and art. So I hope that some of what I'll show today about cells will remind you also of the artistic beauty of life and of biology, which I know attracted many of us to work on these problems. I also have to share my disclosures. I'm a co-founder and equity holder in Celsius Therapeutics and Immunitas. Until July 31st, 2020, I was an SAB member of Thermo Fisher, Neogen, and Syros. And from August 1st, 2020, I'm an employee of Genentech. And I'm going to talk today about cells, which are the basic unit of life. It's not the DNA, it's not the genes, it's actually the cells. And each of us is made of about 37 trillion cells, and they come in many different types, like neurons and immune cells and muscle cells and fat cells. And these cells are different from each other in many different ways. Now, at first glance, you might actually think that every cell in your body should be the same, because after all, they all have the same DNA, and so they have the same set of instructions. But in fact, the cells do have different structures, locations, they do different things in our body. How come? And so let me take you back to the basics of those instructions. Our DNA carries the instructions to make different proteins. And to carry out those instructions, genes in our DNA are transcribed into another kind of molecule that we call messenger RNA. And this molecule is translated to proteins based on the genetic code. Now, while the DNA carries the code or the instructions, the proteins are the business end of the cell. They are actually responsible to executing a lot of its functions. For example, you might have insulin in order to make sure that your cells can get glucose in, or in neurons in your brain, you would have neurotransmitters, or in your muscle cells in the body, there will be fiber proteins that would allow the muscles to contract. Now, every cell in our body doesn't carry just one set of instructions. It actually carries 2 copies of every gene, so 2 copies of instructions. But at every moment in time, it can make no RNA or proteins, so not use a gene at all, or it can use a little, or it can use a lot by making a few or many copies of RNA and proteins based on each piece of these instructions. Now, which part of the instructions a cell will use and to what extent it would do it, that means how much RNA and protein will a cell actually make, depends on what the cell needs. So imagine that there's just 4 genes in your genome, and I'm calling them A, B, C, and D. And there's 3 kinds of cells. There's neurons, immune cells, and muscle cells. These cells need to do different things. Neurons process signals. Immune cells respond to pathogens. Muscle cells contract. And so to do these functions, they need different proteins that are coded by different genes. So for example, cell 1 may need some of A and less of C. So it is going to make a lot of multiple copies of RNA from A, fewer from C, and none from B or D. And I'm going to label how much it expresses in blue. The darker the blue, the more the gene is expressed. Cell 2 may express each of A and B and C and none of D. And so this is going to look like this in my blue colors. And then cell 3 is going to express higher levels of B and of D. Okay, so we call this pattern of how much each gene is expressed an expression profile. And this profile you can think of as the calling card of a cell. Because if we know what the cell uses, then we know what the cell is. Cells that are similar to each other and are doing similar things should have similar calling cards. Neurons all express their neurotransmitters. Immune cells all express other similar genes that they need to fight pathogens. Muscle cells all express fiber proteins, and so on. Now, why does this matter? The fact that all cells in the body have the same instructions, but they use different parts of them, that has critical implications when we think about the genetic causes of disease. If there is a genetic variant that confers risk for disease, all the cells in our body are going to carry the variant. But disease is only going to be manifest in those cell types that use or express the gene. The other cells don't care about the gene and they don't feel the fact that something is wrong with it. And so knowing our cells is essential for understanding how the genes that cause disease act. Now, the only problem is that we don't really know how many different cells there are and who they are. If you open Wikipedia, it's going to tell you that there's about 300 major cell types. But if you ask a neuroscientist, they will tell you that just in your retina, in your eye, there are more than 100 different kinds of neurons, and each of them does something different. And so what we need is a map or an atlas that would have clear coordinates, and it would let us know all of our cells and what they do. But what would be the coordinates or the axes for this kind of map? So the coordinates can be the level of expression of each of the genes. And how would we think about a map like this? Well, imagine for a moment that there's just 3 genes in the genome. We're going to represent each gene by one axis. So X, Y, and Z for genes 1, 2, and 3. And so, for example, this cell that's highlighted with a red circle expresses a lot of gene 3 and very little of gene 1 and gene 2. So it's going to be positioned at this particular point in 3-dimensional space. Now, our genome doesn't have just 3 genes. It has about 20,000 genes. And so there's actually 20,000 coordinates or dimensions in the map. But I can't show you 20,000 dimensions. You can see it with your eyes. But machine learning algorithms can actually handle spaces like that. They can see the map for us. They can discover the right patterns, and then they can show it to us in 2 or 3 dimensions, and they give us these intelligible versions that we can look at as scientists and understand. And I'm going to show you some of these maps in a moment. So this is all wonderful, but how would we get the information for the expression profiles to put the cells in the map? Well, for a long time, we actually couldn't do this for individual cells until new lab methods came along called single-cell genomics. And this is where I will refer to my favorite fruits. So until several years ago, if we wanted to measure an expression profile, know how much RNA we have from each gene, we had to take hundreds of thousands or millions or even billions of cells in one tissue and measure their average. Now, if you think about each type of cell as a kind of fruit and each individual cell as a particular piece of a particular fruit, then what we were doing was basically generating the lab equivalent of a fruit smoothie. In the last several years, there's been major breakthroughs. And in a field called single-cell genomics, we can measure the expression now of genes in each cell separately. And so as a result of that, what we get is a fruit salad we get the individual pieces of the fruit, and so we can distinguish the strawberries from the blueberries, but we can also within the strawberries say, oh, this strawberry is mushier than that one. And you should know that with the latest spatial technologies, they allow us to do the measurements when the cells are still organized together in a tissue. And conveniently enough with my fruits, this extends to the analogy of the fruit tart, if you look at it kind of from the top, and it's a nice start. Today, I'm going to mostly focus on single-cell genomics. How does single-cell genomics work in the lab? One of the key techniques is called single-cell RNA-seq. We start with a piece of tissue, and when the cells are in the tissue, they're actually connected to each other. We dissociate them in the lab in order to make them into a suspension of individual cells. Then we capture each of these cells separately. We tag the cell, and we isolate its RNA molecules that are now tagged by a special tag that tells us that they came from that particular cell. You can think of it as a barcode. Now, after we do this, we can sequence the RNA using sequencers, and that tells us two things: the identity of the genes that the RNA came from, and how many copies of them were expressed in each individual cell based on the barcode. Now, because there are so many cells in the body, we needed lab methods that let us do this very quickly and very cheaply for vast numbers of cells. And so one approach that we developed that allows us to do this is shown in this movie that is taken by a microscope. All of this is very small. The cells get captured in these tiny little droplets in these tiny little devices. And using these massively parallel methods, tens of thousands to even millions of cells can be studied in a day. And this led to an explosion in the number of cells that we can look at. But the question is, of course, what do we learn? And so what we learn is that these profiles of cells can tell us what we're made of, how we develop, and where disease is. And I'm going to highlight this to you through 5 brief stories. The first is about discovering cells in our body that we did not even know existed, and how those cells can be important in a rare genetic disease. This starts with the trachea, which is part of the airways, and at first with a mouse. We wanted to know which cells make the lining of the airways, and so we profiled the RNA of 40,000 cells from the mouse airways with those droplets that you actually just saw. And in this plot, every cell is a dot. I cannot show you the cells in 20,000 dimensions, but our algorithm can take the information from 20,000 dimensions and put it in 2 dimensions, so that if cells were close to each other in 20,000 dimensional space, they're also going to be likely close to each other in this 2-dimensional space. In parallel, another algorithm looked at the high dimensional space for groups of cells that were all close to each other because they had roughly the same calling card. And they colored these dots so that each such group or cluster had a different color. And these could be maybe one type of cell. And in fact, we found in this way that each of the 6 major types of cells that were already known to exist in the airways is reflected by one of these colors, except that in many cases, what we thought was just one kind of cell was actually 2 or 3 subtypes of related cells. But then finally, there was this tiny speckle here in the purple, and it didn't look like anything that anyone has seen before or knew about in the airways. And so we wanted to figure out what these cells might be. And we went to look at their calling card to find which genes are uniquely expressed by these cells, they're in the dark blue, and are not expressed in the other kinds of cells. And based on 3 of these genes, we realized that these cells are similar to cells that were before only seen in the frog skin and the fish gills. And there they were called ionocytes. So we decided to call these the tracheal ionocytes. But also we had a major surprise, because these ionocytes also expressed an extremely famous gene called CFTR, known as the cystic fibrosis gene. This is the gene that has mutations in individuals that have cystic fibrosis. For the past 30 years or so, scientists thought that this cell was actually expressed in another common cell type called the ciliated cells. Those are the cells that are colored red at the top of the plot. But we found out that it is actually expressed in ionocytes and not in ciliated cells at all. Now, because we originally saw this in the mouse airways, and mice are not humans, we then went and looked in hundreds of thousands of cells from human lungs and airways. And again, we found the ionocyte. And we found that the CF gene, CFTR, is expressed in ionocytes and not in ciliated cells. This has big implications. For example, if you want to develop a gene therapy for cystic fibrosis, you need to fix the genes in the right cells. And if you're going to go after the wrong cells, you're not going to end up with effective therapy for patients. Okay. Next, we can ask, where did the cells in our body actually come from? How did they come to be? We all start from just one cell of one kind, the fertilized egg. And yet that one cell begets the many trillions of cells of many different types in the adult body. This process is called differentiation. So again, I'm going to start with an animal model, this time the zebrafish. Now, fish embryos develop very quickly. And by 12 hours, there are dozens of different cell types that are organized in tissues. And so again, we needed to use an algorithm, in this case, another algorithm, to order the cells precisely and recover back this gradually process. They took them from fertilized eggs to these many, many different kinds. And as a result, we found a tree of all of the paths by which cell begets cell and how they diversify and differentiate. We can do this not just for fish, but also for humans. Here is how an algorithm takes hundreds of thousands of cells from the bone marrow. At first, they were all centered in the middle of my slide. And it opens up this picture that traces blood cell formation from stem cells to red blood cells, myeloid cells, and so on. And what is special about these reconstructions is that they allow us to play back the tape. We can look at cells that we would want to obtain, for example, to generate them as sources for therapy for patients, and ask, how did we get there? Which cells did they come from, and through which steps? So here, for example, we're tracing back how we got from skin cells to what we call reprogrammed cells that we can use as the start of many medicines. We can also understand how other cells, like these connective tissue cells, arise in this process at the same time. And these kinds of movies then can give scientists the information that they need in order to engineer cells as therapies. The third vignette I'm going to share to you is about the programs that the cells use. You can think about your cells a little bit like a computer that can run different kinds of programs in parallel in order to do different things. So until now, I talked to you about each of the genes separately, but usually our cells need a whole collection of genes to be expressed together in order to execute a complicated function or a program. So the expression of these genes rises and falls in a coordinated way. And as a result, that's why we call them programs or modules. Even cells of the same type would need to shift the gene programs that they use in order to fit their circumstances. And this is especially important in diseases, including in cancer. So for example, some of you might have heard about therapies called checkpoint inhibitors that have had huge benefits in cancer care. As a brief explanation, immune cells that are called T cells can identify and kill cancer cells. But cancer cells are nifty and shifty, and they can avoid being killed by T cells by hijacking one of the natural ways in which our body shuts off the immune system when it doesn't need it. And when the cancer cells activate this, they shut off the T cells, they inhibit them, and sometimes we say that they exhaust them. Now, there are medicines called checkpoint inhibitors that cut this off. And as a result, they unleash back the T cells, which can then come in and attack and kill the tumor. This has brought amazing benefits to many patients, but in other patients, they do not seem to work. And we say that those patients are resistant to immunotherapy. So now we can ask, what is the difference between the cancer cells from tumors that are resistant to immunotherapy and the cancer cells, still cancer cells, in tumors that are responsive to immunotherapy? They're all cancer cells, but they're somehow different. For this, we looked at the cells not from healthy tissue, but from patient tumors. And we compared tumors from patients that have not been treated yet to those that have failed their immunotherapy. And again, the dots are our cells. In the top plot, they are colored by patient. Tumors are very different, so each patient's cells separate from each other. In the bottom plot, we color them by their treatment. But in this third plot, they're colored by the expression of a program that we discovered, a set of genes that are used by cells in cold tumors that don't have T cells in them and so are resistant to immunotherapy. In general, we see this program active in cancer cells in the patients that failed treatment. But it is also present in some of the cancer cells of tumors of some patients that have not yet been treated at all. And this would suggest that patients like these might not benefit from immunotherapy. And indeed, when we looked at tumor cells before therapy from a large cohort of 100 patients, we found that this program is present in the tumors of patients that are less likely to respond to immunotherapy. And so we can now predict which patients might respond and might not. Now we, of course, would ask, can we find a way to reverse this program? And we found a family of drugs that when they're given to cells that have this exclusion program that throws the T cells out of the tumor, it can revert them back to the sensitive state. And so now we can propose a new option for treatment, combine the immunotherapy with the additional drug. The drug would make the resistant tumors hospitable to T cells, and the immunotherapy will allow the T cells to kill the sensitive cells. We've already shown that this combination is successful in animal models, and our clinical colleagues are exploring this in patients. So finally, cells tend to reuse this program. It's like little code pieces that you can use in multiple places, even across diseases, and in this case, across tumors. So when we look at this program across the many tumors in the Cancer Genome Atlas, or TCGA, we see that on average, this program is lower in cutaneous melanoma, lung adenocarcinoma, and kidney cancer, so cancers of the skin, the lung, and the kidney. And in fact, patients with these kinds of tumors are known to benefit more from immunotherapy. On the other hand, cancers like uveal melanoma, it's melanoma of the eye, and colon cancer, all have relatively high scores. And patients with these kinds of tumors, on average, benefit less from immunotherapy. And the highest score was in synovial sarcoma, which is a cancer of young adults. And this led us to discover a related program in the cells of synovial sarcoma. And it is present in some of the cells of every synovial sarcoma tumor that we studied. Patients with high expression of this program have worse outcomes. And this program is controlled by the master driver of the tumor, and it leads to T cell exclusion. And this also identified a potential treatment combination for patients. So we looked at rare disease, we looked at regenerative medicine, we looked at cancer. The fourth story that I'm going to share with you is about a common disease with a genetic basis called ulcerative colitis, which is a form of inflammatory bowel disease, or IBD. Now, human genetics has been very effective in IBD in finding new genetic change, in making new findings of genetic changes that are causes of disease risk. And they can highlight areas to develop new drugs. But unlike diseases like cystic fibrosis that are caused by rare mutations in just one gene, the CFTR gene, here there are hundreds of genes that have variants that are present in many people. They're not rare, they're common. None of these genes and variants on their own would cause the disease alone, but specific variants increase the risk. And for the vast majority of those genes, we do not really know what they do at all. And so how do we proceed? So to help answer this, we made a map of the cells in the colon of healthy individuals and of patients with colitis. And just like before, each cell is a dot, each color is a type. Now there are over 50 different types and subtypes of cells, and the algorithm organized it nicely on the face of a sphere to help us see it better. Some of these cells are present only in the diseased tissue, and we do not see them at all or see very few of them in the healthy patients. And these cells that emerge in the diseased tissue also form new connections in the disease. So now we can hypothesize that maybe targeting these cells and connections would be beneficial for treating patients. Next, we can ask, for each of those hundreds of genes that were related to the risk of ulcerative colitis, which cell expresses it? Here, red means high and blue means low. And you can see that most of the risk genes have these individual red spots. And so they're expressed or used by only very specific types of cells. And even though there's hundreds of genes and dozens of cell types, groups of these genes go up and down together in programs within specific cell types. So for example, a rare cell type, which we see only in disease, called M cells, has 4 such coordinated risk genes. Now, each such group of genes in a specific cell type can be a target for therapeutics. Now, the map of the cells and the gene programs allow us to compare better between related but different diseases. In the case of inflammatory bowel disease, in addition to the common form, where there are many genes, each of them with variants that are common in people, contributing a little bit to the risk of disease, there's also what we call monogenic forms of the disease. That's similar to what I showed you with cystic fibrosis in the lung. It's just one gene with a major genetic defect that leads to very severe disease, usually very early in life, in young children. So in some cases, the defects end up being in the same genes. It's just a different level. There are 13 such genes. But when we ask which cells or programs the genes are expressed in, we have a much higher overlap. So even when it's different genes, these seemingly different diseases actually converge to the same cells and programs, which suggests that we could develop the same therapies. And finally, this map was useful not just for ulcerative colitis or IBD, but to understand genes for many diseases that we know affect the function of our gut, including rare diseases like Hirschsprung's, where the neurons in our gut don't work correctly, but also neurodevelopmental and neurodegenerative disorders like autism and Parkinson's disease, where there are also gut symptoms. And so I showed you 4 stories so far on how mapping cells can give us insights to rare disease, regenerative medicine, cancer, and common disease. But with all of this knowledge in hand, can we also understand how the genes cause the disease, not just where they are? And for this, we want to use one of the greatest superpowers of biology, genetics. In biology, we can actually go into the genome and perturb the genes and see what happens. And because the genes are the starting point, we can infer that the gene causes the effect or the phenotype that we see. We can do it in one or two ways. We can rely on natural genetic variation. Each of us is genetically different than each other. And we do this by looking simply at different individuals. Or in the lab, we can introduce perturbations by engineering them into cells using experimental tools. In each of these cases, to see what happens, we can now use our new lens of single-cell genomics to look at the effect on cells. And I'll show it to you with just one example, where we wanted to understand the function of 35 genes, each of which was found to have a new mutation in a child with autism, where neither parent has the mutation. And so we think that this mutation abrogates a gene's function and contributes to autism. So here we know that each of the 35 genes is causal in autism, but we don't know how that happens. We don't know in which cell or process it participates. And so what we did was to deliver a pool of ways of perturbing the genome, in this case of mice, to disrupt each of the 35 genes in mouse embryos in utero, when they were still in the uterus, and then recover the cells and profile their RNA after the mice are born. And when we characterize the effect of the perturbations on major cell types, 5 major cell types in the brain, only one of the 35 genes had a significant effect on the proportion of cells of major cell types. It increased one kind called oligodendrocytes, and it reduced another kind called microglia. But when we turned, like I showed you in melanoma, to the level of gene programs, things changed. So let me first show you what programs look like in cells called projection neurons. Some programs, like PN1, reflect a subtype of a cell. Here, these are called the layer 4 projection neurons because your cortex has layers. And they express this program in red. But other programs, like PN2, are reused in some of the cells of each type. And here, this highlights a process called neurite development, which is something that some cells of each type need to do. And 15 of the 35 autism genes affect 6 of these programs across 4 cell types. And this means that there are likely only a limited number of cellular processes that these genes converge into. And also that perturbation in one gene can impact programs in multiple cell types. And you know, each of these genes is a human story and an individual. And we hope that this map will help guide scientists that are focused on therapies for those individuals. And so if having a map is so much better and so useful, why not make an atlas of all human cells? As a comprehensive reference map of the types and properties of all human cells that we can then use as the basis to understand and diagnose and monitor and treat in health and disease. And this became the mission of an initiative called the Human Cell Atlas, and we sometimes refer to it fondly as the periodic table of our cells. But humans and their cells are diverse, and scientists are diverse too, in who they are, in where they live, in where they came from. And so this should not be something that one lab or one institute or even one country builds alone. And so together with Sarah Teichmann, my wonderful colleague and friend, in 2016, with 93 of our best colleagues, we launched an international planning process for a human cell atlas. And a year later, in 2017, this initiative started its full-fledged data collection. And the International Human Cell Atlas Initiative is rapidly growing. We have more than 2,200 members from 77 countries and over 1,100 institutions. It's open to anyone who is willing to adhere to its mission and values, which include scientific rigor and innovation and excellence, but also commitment to equity, diversity, and inclusion in both research subjects and the scientists, rigorous ethical standards for our patients' consents and privacy, as well as transparency and open sharing of our data code and lab methods. The Atlas is rapidly growing. HCA members so far profiled almost 55 million cells from 18 organs and systems from almost 8,000 individuals. And in addition, HCA scientists have been committed to contribute to the fight against COVID-19 and have studied more than 11 million COVID-19 patient cells from multiple organs and from the blood of almost 900 individuals. Just as one example of this dedicated effort was our assembly of atlases of organs from autopsies of patients who tragically succumbed to COVID-19. And here I'm showing one of these largest atlases that span the lung, the heart, the liver, and the kidney from 17 patients. By analyzing this data, not just on its own, but in reference to the healthy atlas of the same organs, we're able to show the dramatic changes that happen in cell composition in each organ. We could see how COVID-19 in the lung leads to many failed attempts of the lung to regenerate and heal itself. We could determine which cells are enriched for infecting virus and what changes this infection causes to their programs. And we could relate the cells to genetic variants that are associated with risk of developing more severe COVID-19. And we hope that all of those would help scientists and doctors understand the disease and help patients. Overall, we expect the Human Cell Atlas to impact many aspects of understanding and treatment of disease. I showed you today examples for diagnosis and prognosis in cancer, of human genetics and IBD, of regenerative biology and cell therapy, and of COVID-19. And it's a great pleasure for me that both Genevieve Almuzni and Nikolaus Rajewski, who are a leader of an initiative focused on disease lifetime, are actually in this audience today. We rely on you. And we are well on our way to hit our first draft at our 5-year mark, and we're working towards an even more comprehensive and complete atlas in our decade-long effort. And with this, I will conclude. I will thank you very much for this great honor, and I am thrilled to be here today, even if only virtually. Thank you very much.
Thank you very much for this prize lecture, Aviv Regev. And you have the opportunity to ask her questions now. Did you understand everything? You can ask her. I didn't understand everything, therefore I would like to ask one question. You said the cells are like a computer that are able to run different programs. But how does the cell know which program to run?
Oh, that's the million-dollar question.
Okay, sorry.
No, it's a great question. So the cells constantly receive signals. They have a lot of sensors. They have sensors of the outside world. These are proteins that they put on their cell membrane and that are receptive to signals from the outside. And they also have sensors internally that are receptive to their own situation. And based on all of the signals that they receive, they execute a series of events that all go through these molecules. One molecule talks to a second molecule, talks to a third molecule inside the cell. And many of those, although not all, but for simplicity, many of those end up in the cell nucleus where the DNA and the instructions sit. And as a result of that series of events, particular proteins called transcription factors will go and they will actually bind the DNA. And they will either repress or activate the expression of genes. So it's a kind of a self-perpetuating thing. It starts, as a fertilized egg, as I said. And at first it has this internal drive to change itself, but it's always feeling what's around it. What are the other cells doing? What did I just drink? What did I just eat? And so on. And all of that information gets sensed by the cell, percolated through these, we call them circuits. A little bit like computer circuits, where molecule tells something to another molecule, tells something to another molecules, and cells manage to implement through all of these proteins talking to other proteins, pretty much every sophisticated function that you can imagine, including all these wonderful things that our brain manages to compute without us even giving it a second thought. So it's a pretty remarkable set of abilities that cells have. Which is why they are the fundamental unit of life. You cannot reduce them further.
When you talk about cells, it sounds like you are talking about good friends. When did this fascination about cells start? Do you remember?
So I actually didn't even think I would study biology at all. Didn't in high school. I'm Israeli. We go to military service, then we go to university. Couldn't exactly choose what I wanted. And I stumbled into a genetic class in my first semester that was taught in a very non-traditional way. Let's leave it at that. But genetics, as you know, is actually a very mathematical way of thinking about biology. You do things by inference. You observe the outcome from perturbations and you try to work backwards, reverse engineer what might have been the system that led to what you're seeing now. It's like taking your cell phone, opening it, cutting a wire, seeing if it still rings, something along those lines. And I was really fascinated by that. Took a second class, which was also very mathematical in evolutionary biology. The same story, there's lots of math and theory and how you think about it. And then I was like, oh, biology, that's amazing. And I took a molecular cell biology class, which is a much more typical place to start biology. And people talked about these stories, right? Like the one I just said, there's this molecule and it does this to that molecule and it does this to this other molecule. And kind of two things occurred to me at once. The first one, there was no math, there was no computation, none of those things were part of it. People just talked about the lab findings, and I felt that was missing. It was hard to put the pieces together and really understand the principles. But on the flip side of it was this richness, like almost this unbounded richness of what cells and molecules can do. And so I felt, oh, that's a good problem to work on. And I still do. I guess it was a good choice.
It was. Thank you very much. So, any questions? Stefan Kaufmann, we have an extra microphone. I don't know if... yeah, you...
Is mine working? Or?
Yes? No?
Thank you, wonderful talk. I was wondering whether you would also consider, first, this is an enormous progress, and I think this is a lot of work already now, I know. But would you also consider the microbiome in there? Wouldn't it be also interesting to look at that? IBD, for example, but also some neuro diseases, that it's not only the different cells, which are as many as our own body has, but also on their status, I would assume. It's not only, as most people think currently, the kind of composition, but also the activation status of those cells in the microbiome.
I agree 100%. And actually, we have spent a substantial amount of time trying to get over a technical barrier in order to have simultaneous measurement of both. So first of all, I didn't get to this in this talk, but the work that we've done was with a cohort from MGH, with my colleague Ramnik Xavier. And in fact, for that same cohort, there is also microbiome data. But we always had to look at them separately and then try to find computational ways to try and bring things together. For example, identify molecules that the microbiome makes, and then find ways to understand how they converse with the cells that we see in the body. And one area that we realized was actually a technical barrier for us. A lot of this interaction between the microbiome is actually from cells from the enteric nervous system. For those in the audience not familiar with it, the second place in the body with the most neurons is your gastrointestinal tract. It has 1/200 the number of neurons in your brain. And that's a lot of neurons. And so that's something unique to the gut, and those cells were very difficult to profile. We actually had a dedicated study with multiple lab methods we had to develop in order to get to those cells. Now that we have them, we can understand the interaction with the microbiome a lot better. The second barrier we wanted to pass was to actually be able to profile the microbes simultaneously with other cells. And that introduces a whole set of issues because the microbial cells are so much smaller. They have a cell wall that makes it harder to dissociate and get into their contents. And so our focus has been after some failed attempts, our focus has been primarily on spatial methods and using the fruit tart approach. And we do have a technique for that is an extension of a technique called spatial transcriptomics that allows us now to look both at the microbial side and the host side simultaneously. And so we don't just get to say these microbes are there, but we get to say these microbes are there next to these host cells. And we hope from that to back up the conversation that happens between them.
Thank you very much. Are there any other questions? We have a microphone here. Just give a hand over there, a young woman. Okay. Wait, the microphone is there.
Hello. My question is if the cell atlas only shows specialized cells or also multipotent stem cells already?
Yeah, so one of the big efforts two relevant efforts in the Human Cell Atlas, one is the developmental atlas, which is part of the Human Cell Atlas that starts prenatally and also includes a pediatric atlas effort now to look at young children, which is a separate and related category. And so there is definitely information from the stem cell dimension from that side, which is a long process of development for humans, and we get certain glimpses into it with some obvious limitations. The second part is that there is a large effort and a network in the Human Cell Atlas focused on organoids, which is a way in which we can look at stem cells in a model system, but based on human cells. So one of the great advances that has happened in the last 15 years or so allows us to take human cells and actually grow like almost a little organoid in a dish. And you could do it, originally this was done with gut organoids, then now with many, many other organs, even brain organoids. And you can do it from adult stem cells, and you can also do it from pluripotent stem cells, depending on the system that you're striving for. So the organoid network really focuses on that, and between the developmental network and the organoid network, we feel that we have a good way of triangulating around those cells. I'll add that the technique I showed at the very end with the mechanistic work that you can add genetic perturbations is going to be particularly useful for questions in regenerative medicine and in stem cell biology, because you can introduce a large number of perturbations in those systems, and then you can monitor what happens to cell differentiation after.
Thank you very much. Are there any other questions? in the audience. Oh, here too, in the front row. Okay, the microphone is coming.
Thank you for this outstanding description of the paradigm of the cell atlas. So every cell could be in a different state, activated, exhausted, and so on. So this will add an unknown number of dimensions to the cell atlas. So could you give us some thoughts about the future paradigms of the cell atlas?
So this is actually why I spent a good amount of time talking about programs and not just about types of cells. So you could imagine a situation which is almost unbounded, because at any moment in time, say the cells in my body, I just took a sip of coffee, they're not exactly the same as they were 10 minutes ago, because they were, at least some of them were responding to that signal. That's true. But they're responding, we believe, from a limited vocabulary of programs. And what they do is actually combine those different programs to form their state. So their type is characterized by one very substantial program usually, which is quite invariant with time and is stable, and you can see very quickly. But their states their overall state is not just reflective of their type, but of the many other programs that are active in them. If the world were really simple, then those things would just be discrete units separable from each other, and we just add them up. Biology is never as accommodating as that, because there are non-linear interactions between those things. But still, the vocabulary of these programs is going to be both one of the outcomes of the Human Cell Atlas. It's not just the catalog of cells, but it's also the programs that they run on. And it's also one of the things that then gives us a limited set of units, maybe unbounded or very large in the different number of ways in which they can be combined, but the building blocks are manageable. And that's I think, where we're headed. Biology in general, for those who come from the mathematical mindset, tends to appear very unbounded in principle, meaning it's an unimaginable number of how many combinations, for example, could exist, and more than the number of atoms in the universe and so on. But if you think about which ones are actually realistically there, you're usually working with a much smaller number of dimensions. This is why you can take a 20,000 a 20,000 dimensional map and reduce it to 3 dimensions and still maintain most of the relationships that were there. It's because in a deep way, there aren't really 20,000 independent things going on. There's a lot of relationships and dependencies between them.
Thank you very much. Another question in the front row here?
So this was an outstanding talk. Thank you very much. I was wondering, so single-cell genomics has only been there for a couple of years. Bulk sequencing has been there for decades, right? And so there's an immense amount of data out there. With all the new information that you gain from these single-cell genomics, can you go back in time and make these smoothies, as you call them, more towards a fruit salad by reanalyzing them?
Yeah, it's a fantastic question. And yes, you can back out the smoothie. You can even do more than that. I'll tell you in a second. So first of all, it's been around for more than 2 years. And the initial work on the methods comes from the early 2010s. I would say there was works in 2011, 2012. There was even work earlier than that, but the scale was so small that it would be like one cell and very noisy. And if you really want to give credit to the beginnings of single-cell genomics, you have to go back to the 80s and the 90s, where people would take single cells and analyze them with Sanger sequencing. So people have tried many times, I think, because they knew it would be valuable if it succeeded, but sometimes you have to get over the technical barrier, and in this case, also the analytical computational barrier. So it's been around for a while, and I would say in the last 4 or 5 years, we're seeing the really broad adoption. Before then, it was more for the aficionados. Now it's a tool in the biologist toolbox, and it's a conceptual way in which biologists work. Now, to the question on backing out the fruit smoothie, absolutely. And we have done that. We have done that in cancers. We have done that in the example I showed for IBD, for inflammatory bowel disease. We actually then took this 51 cell type atlas, and we reapplied it to biopsies that were obtained from patients that before and after treatment with anti-TNF therapy, which is a very common therapy for IBD. And we were able to back out the smoothie, and we were able to understand which cells are actually contributing to resistance to anti-TNF therapy. And that was very useful because some of these cells were cells we didn't actually know existed in the gut, or states of cells, in this case, inflammatory fibroblasts, that we weren't aware of before, and they're rare, and you wouldn't be able to see them unless you know how to back it up. It's still a lot of work to do the backup, and we are getting better and better at it. There's other things that we can now do. For example, one of the most, the oldest techniques for single cell analysis has been H&E stains. This is, I think, 150 years old or so, definitely 100 years old. No, more than 100 years old by now. It's 2021. And it's used every day in clinical pathology in order to stain tissue and look, and you can see cell shapes there, you just don't see the molecules. Well, it turns out that if an algorithm today looks at enough examples of an H&E stain and a single cell RNA-seq dataset without any molecules measured in the spatial domain, it can be given another H&E stain and generate a single cell RNA-seq from it. It literally can generate a fruit salad, and then it can put them on the spatial image, and it can generate a fruit tart. So almost like give you the technicolor from the black and white, which I'm sure you've seen all these crazy things where people take a black and white photo from 1901 and they color them. That can be done now when the colors are molecules. So there's a lot that can be done with the new data, not just to collect more new data, but also to go to older legacy data that was hard won and make it all of a sudden in Technicolor.
Thank you very, very much, Aviv Regev, for your lecture, for answering the questions, and for your work, for the fruits, and for your enthusiasm. Thank you very much for listening and for your questions here in the audience. And I warmly and deeply recommend the graphic novel about Aviv Regev's work from author and illustrator Jan Feind. You find a lot of pictures and speech bubbles, and you can even more understand about this theory of fruit salad and smoothie. Thank you very much. We have a small break now. After 15 minutes, the prize ceremony continues at 6:30 p.m. Thank you for joining Aviv Regev.
Thank you.